Hi, everyone. Welcome to this short video case study that we've put together about one of our neurodegenerative disease testing programs in Parkinson's disease. I'm the co-founder and CEO of Sano Genetics. My name is Patrick Short. My background is in rare disease diagnostics and population genomics. And I'm going to spend about 15 minutes taking you through a short case study here based on some of the past work that we've done in Parkinson's disease. So the purpose of this program was to find and screen and engage patients with genetic forms of Parkinson's disease to build a cohort for natural history studies, clinical trial enrollment, and help a venture-backed biotech to accelerate their clinical development in this traditionally hard to reach population. And I'll go into some of the details in a moment. So some of the challenges here, why was this a difficult study in the first place? In particular, it was multi-country, multi-language. We needed to find and genetically screen patients both in the US and Europe that had a rare genetic subtype. If you're not familiar with the genetic epidemiology of Parkinson's disease, there's a number of genetic drivers, including LRRK2, GBA, and some rarer genetic variants. And increasingly, people are targeting these genetic variants in clinical trials. So it's important that this population was identified not just as having a diagnosis of the disease, but also having the right genetic subtype to take part in subsequent trials. And then once patients were screened, they needed to be retained until the appropriate clinical trial or natural history study became available to them. So as you can imagine from the genetics I described previously, some patients would come through the process and find out that they had a LRRK2 variant, others would find they had a GBA variant. And the purpose of running this kind of umbrella screening program was to more effectively and efficiently screen patients rather than doing this one by one as trials came up, which would result in very high screen failure rates and spending a lot more money on recruitment. Instead, this could be funneled through one core umbrella screening program that allowed patients to have a much better experience by getting access to more comprehensive testing. And also, while preparing and enrolling for one trial, you can also prepare and enroll for upcoming trials based on additional genetic targets. So for the particular genetic target we were going after in the first instance, we knew that there was a approximate prevalence of 3%, just over 3% in previous genetic testing programs. And this number is important to keep in mind for two reasons. First is this represents a really core part of the screen failure rate in a traditional trial context. So if you wait until Parkinson's patients, in this case, arrive at a clinical trial site and get genetically tested there, you'd expect almost 97% of them would screen out just on the genetics alone, not to mention all the other reasons that someone will screen fail out. And so you can imagine that the screen failure rate is dramatic in this case if you do the genetic testing solely at clinical trial sites. This is important for a second reason, which is that this is a number that's been achieved by previous programs, but it doesn't mean that it's the best that a program can do. One of the things that makes us unique as a company is because we're actually not a commercial lab. We don't make money on the volume of tests that we do. We make money by running a program that's efficient and optimized and gets you to the end goal more quickly and effectively. And so for that reason, we're able to think about how do we actually increase this hit rate by designing better pre-screening questionnaires, by looking at recruitment channels that are delivering more effectively because people are more willing and interested to take part, or for designing recruitment materials that appeal a little bit better to the target population, which in this case may be people with a family history, for example, who are more likely to test positive. So those are a few of the examples of optimizations we've made through to get to a much higher hit rate than what we would consider an industry standard before we ran this study. Some other challenges here is we know that patients lean on their healthcare professionals for decision making in clinical trials and more broadly around health. And this is something that we don't want to take away or replace, but we also need to acknowledge that not every neurologist, not every movement disorder clinic has a full understanding of genetics and genetic counseling and the full range of clinical trials that might be available for their patients if they had access to genetic testing and counseling. So this was really a partnership-driven approach where we were allowing patients to take part in a study like this directly, but also working really closely with healthcare providers to give them access to the resources that our platform provides, like free testing and counseling and educational materials that help them learn more about why genetic testing is important, why the industry has changed and is changing, and why it's going to become an increasingly important part of their healthcare practice in the future. So what was the approach here? In brief, we allowed participants to take part through our online screening platform, and if they were eligible, having a diagnosis of Parkinson's disease and meeting some of the other required criteria, then they would get sent a non-invasive at-home DNA test. So this is a saliva-based test that allows people to take part in 5 or 10 minutes from home. It's very easy to do, and shipping is handled to and from the participant's home, all inside the kit. This can also be sent to clinical trial sites or partnered healthcare providers as well. So it doesn't need to just be direct to patient, but can also go to healthcare providers who can talk through the genetic testing with their participants and help them understand what the impact or implications of this might be. And ultimately, the impact of doing this kind of non-invasive, easy at-home test means you can reach a much wider range of patients. They don't need to travel to take part. It's non-invasive, so people, if they're concerned about doing another blood test, they don't have to worry about that. And ultimately, this results in lower patient burden and lower recruitment costs because you can make it accessible to almost anyone. We further partnered with a healthcare market research company in order to reach out to not just the healthcare practices that were key opinion leaders and those who see, lots of patients and may be familiar with genetic testing, very familiar in some cases, but also, and in this case, more importantly, reaching out to all the clinics who don't necessarily have the key opinion leaders that are publishing and writing about genetics every day, but are still seeing, in some cases, hundreds of patients that are really interested in getting involved in research and really interested in getting access to comprehensive testing for their patients. So we found it was really successful actually to focus on not just the top tier key opinion leader sites, but also sites that had real interest and drive to take part in research and many interested and motivated patients, but may not, for example, already have access to testing through their academic medical center. And so we found this was a really key insight and applies to almost every program where you want to, of course, work with the experienced healthcare providers and sites, but also you don't want to ignore the fact that there are many great sites that don't have the resources that those extremely experienced ones do, that a program like this can help get them involved in research and accelerate the progress of the trial for you and for your patients. Once someone is eligible, after they've completed the pre-screener and they've completed their tests, then they get access to what we call a virtual waiting room. So this is a personalized digital space for the participant that allows them to understand more about their genetics, underpinning not just their disease, but other traits or educational information that might be useful to them. It also includes ways to learn about new research opportunities, whether that's clinical trials or pulse surveys that are helping you understand something a little bit more about the patient experience, how they might react to a protocol or a certain kind of study design. So this is a really important part of the journey, in particular because we know many trials are long running and participants like to have a good understanding of how things are going, but also there may be a wait between getting a genetic test result back and actually finding a trial that is relevant for them. As I mentioned at the top of the presentation, we don't know what genetic result a patient is going to have when they start this process, and it may be that there's a trial ready to go today for them, but it may also be that the trial that's right for them is not planned for a year. And so making sure they understand that there's a trial coming, but it's not quite live yet, it helps them to understand and prepare and then be able to make that decision in a more informed way when the time comes. From a clinical development perspective or clinical operations perspective, we set these up in a pretty unique way. We call them patient finding protocols. So they sit independently of any clinical trial protocol, and they're really focused around genetic testing, education, and building a cohort of engaged and research-ready patients that sit in this separate protocol. And the reason why it's important to have it separate is because we can make changes and innovate and do different things in this protocol without having to touch the trial protocol, which obviously serves a different purpose. It's for testing a new medicine, and we want to separate these two because it allows us to move faster and be a little bit more innovative. So the virtual waiting room I just described sits on top of this patient finding protocol. So when people consent to join the patient finding protocol, they join this virtual waiting room platform. And that is where any IRB approved recruitment materials for this study or for future studies can be shared with participants, not just in the digital platform, but also, of course, via email, SMS, and we have members of our team that serve as white glove concierge service as well to reach out to patients individually to help ask to help answer any questions that they may ask. And then finally here, you can think about each step of a clinical development process. You're able to tap into this patient finding protocol and virtual waiting room. So if there's a phase 2 or a phase 3 trial that someone is immediately eligible for today, then of course they're going to want to be interested in learning more about what that involves. But likewise, if they carry a genetic variant that maybe there is not a trial that's relevant for them today, there's still going to be natural history studies and earlier stage biomarker development where their surveys, their samples, or their physical participation at a site may help. And so by structuring it this way, you get a lot more efficiency out of your recruitment efforts. Instead of being narrowly focused on enrolling a phase 2, for example, and telling every patient that doesn't qualify that you're really sorry and losing track of them, actually you're able to present a much broader offering to anybody with the disease who's interested in taking part, and that helps everyone to just run more efficient research and get recontacted when there is something that might be a better fit for them. So what were the results of the program? Over the course of a little over a year, we tested more than 1,000 participants, and we were, expecting these participants to come from a wide variety of channels, and they did. This includes advocacy groups, social media. It also includes healthcare providers, both key opinion leader sites and up and coming researchers or even research naive sites that were interested in learning more about how genetics may change their practice. We were able to increase the hit rate from the 3.1% that I mentioned at the start of the call to over 6%. And this was due to optimizations of the pre-screener, improvements of how we did outreach and recruitment, and a number of other changes. We made more than 20 different changes throughout the program in order to continue to improve this hit rate. And as I mentioned before, this is really a unique aspect of the way that we work. Because we don't make money on simply driving test volume, we're going to be partners with you and your co-pilot in figuring out how do we run the most efficient and effective program overall. So as a result, we had about a twofold greater success rate with finding patients with this particular genetic subtype that we were focused on, as well as others with other genetic subtypes that take part in future research opportunities. We saved about 55% of the cost using the at-home DNA testing service. This is not just for the blood versus saliva itself, but you can also think about additional cost savings that aren't captured here, like travel to and from the site. And also there's an overall cost saving to the program because we know that patients are lost when they're required to travel and have to fly in some cases to take part in research. We want to minimize that burden as much as possible. And this was it made a business case for expansion to new geographies because it was so successful in the geographies that we initiated it in. So that's a brief overview of this case study. We're obviously happy to talk about this and any other programs in more detail. If you're running a clinical trial or natural history study, or in the early stages thinking about your clinical development program for genetic rare diseases, genetic subtypes of common disease, or other complex biomarker-driven studies, and we'd really love to speak with you. We've got a lot of experience now running, dozens of different programs under this umbrella screening model that I've described. So we have a great, platform that's set up to deliver these kind of studies and help you run your programs faster, help you run them more efficiently, and deliver a great experience to your participants. So I'll end here. I'd like to say thank you for your time, making it to the end, and please feel free to email us anytime at hello@sanogenetics.com, and we'd be really happy to set up a call and see how we can help.